Hi, my name is Joshua. I'm a student here at the University of Guelph, and as Tammy said, our lab tends to it's based in MCB, and we study drug-resistant microbial pathogens. And what I kind of want to talk to you guys today is a little bit about the immune system and the interplay that exists between both it and these pathogens, and what we're really doing about the antimicrobial resistance crisis. So, before I get into the crisis, let's kind of take a little step from where we came from. So these pathogens have caused diseases, numerous diseases throughout time. We have cases where we've had the Great Plague, Great Pestilence, and cholera outbreaks, and we usually thought that they were caused by some disease, like ghosts or something wafting through, touching us, or ill wishes. And upon a literal close inspection, we see distinct microbial pathogens just kind of being the causative agents of these problems. So we have cases where you have Yersinia, Salmonella, and cholera causing mass diseases, and the reasons that they were problems is that they infected us and spread so quickly that most humans can really cope with it. So our immune system, because you guys are getting into that, actually does a really good job of handling microbial pathogens. What happens is that traditionally an immune cell will sense the presence of a pathogen, and this is called a phagocyte, and then it puts it in something called a phagosome. And as a phagosome matures with a microbial cell in it, it secretes these enzymes, and it lowers the pH and does all these things to degrade the biological material present. Now, the pathogens and the diseases that I mentioned earlier were so successful, not because of anything novel that they're doing, aside from the fact that they just have really great mechanisms of evading our immune system. So they would get phagocytosed, and then they would actually secrete these molecules that would break out of the phagosome, and it actually also had mechanisms of just escaping the cell as a whole. So how do we get really from millions dying to not that many dying from these old world diseases as we think of them? Well, we invented antibiotics in the 1950s. And they kind of did an amazing job for us. And what they kind of did was just kill the cell beforehand. And once the cell was dead, all the immune cells really had to do was go about and just pick up and break down the dead material, so that it no longer really had DNA or anything bad that can affect us. And going into just kind of how antibiotics work briefly, because we're talking about antibiotic resistance, it's just we develop these compounds that can target the cell wall and cell membrane, can target actually the DNA or the RNA present in these microbes, can actually target specific proteins or the ribosomes that make proteins. And these are all compounds that target the microbial cell really well. They target its metabolism and obviously induces a very strong selection pressure. And we know from kind of classic evolution, any microbe that kind of has any genetic mutations that allows them to survive are the ones that will survive, and they end up with modifications that range from blocking the drug from entering the cell, actually modifying the protein target as a whole, or even just sensing the presence of the drug and just kind of quickly taking it out of the cell before it can do any harm. Now, these problems and these mechanisms of drug resistance are quickly spreading throughout the world. I mean, the WHO has predicted by 2050 over 10 million people will be dead due to drug-resistant pathogens. And something I want to highlight here for you guys is this is not just kind of any pathogens. These pathogens usually have specific mechanisms of evading our immune system, which is why they became a problem in the first place. And so we went from having drugs that kind of saved us from these infectious diseases to a state when now they're becoming a real looming threat. And 2015 might seem really far off for you guys, but it's a very short amount of time, especially given the fact that it's been around 60 years since we have actually invented drugs, and we've reached this new age of hygiene or lack of these diseases, as we'd like to think. So tying it back again into the research that I'm doing and what we're actually looking at, I wanted to show you guys the pathogen that we work with in our lab called Candida albicans. And in this video, I'm going to show you it's kind of already been phagocytosed by a mast cell, which is just a different type of immune cell. And this is actually a drug-resistant version of the strain. And I would like you to see that in the video, actually signals such as the lower pH signals it to form hyphae, which are these long filaments that actually allow it to break out of the immune cell. And then once they form filaments, they start to form these very dense networks known as biofilms that kind of like cover and hide the cells from like other immune cells. And what this means is that these cells the immune cells can't actually penetrate these like thick layers anymore, and they become these huge swaths of persistent and chronic infections, especially amongst those that are immunocompromised in a hospital. And so I'm here kind of like talking about the fact that we have these drugs, they now like don't work, our immune system's kind of like back to square one with dealing with these diseases, which means actually that they don't even really deal with these diseases, and so we're running into a problem. And the solution that research is kind of like looking at right now are these compounds called antivirulence compounds. So what that kind of means is before we had ways of just kind of like killing the cell, and so our immune system just had to do like the job of picking it up. But we're looking at ways of actually kind of like stopping specific mechanisms that these pathogens use to either evade our immune system or actually become virulent as a whole, and just kind of like stopping them from doing these processes. And this way our immune system can kind of like clean up and do its proper job. So take a case here with Candida albicans. You can see like a wild type strain there forming hyphae, like all well and good. And with the induction of a compound, just called 902, we'll say, like at very low volumes, you can see that it's completely inhibiting its capacity to form hyphae. Not only is it inhibiting its capacity to form hyphae, what this means is that it's locking the pathogen into like its yeast state. And we actually see when we have like Candida albicans under huge different types of media that are supposed to imitate like situations in the body, it forms hyphae normally, but with the addition of 902, it completely abolishes hypoformation. And this is actually really important for multiple reasons. So we have cases where it can't really form biofilms anymore, because you can see in the untreated cells, these huge dense networks that immune cells can't penetrate, but then when they are treated, like you can like just see like these sparse like cells, and what actually happens as well is like when they form hyphae, they actually like kind of like send signals to divide, and we don't see that much anymore. And what this means is we can increase the survival rate, and this graph kind of shows that. So in a control where like they are infected with Candida albicans, and these are mice, they're infected with Candida albicans, you can see like after like day 4, like they start to die like very readily. And those treated with this compound 902, like you can see they have close to like an 80 to 90% survival rate. And all that's really happening is that because the yeasts are locked into like this yeast state, when the immune cells actually phagocytose them to actually start to degrade them, break them down, usually they would break up, and you can see that in the control group. But with the ones treated with 902, they are not breaking up any longer, and so the immune system just kind of is allowed to do what it's intended to do. And we're actually doing this for whole swaths of diseases that are becoming very drug resistant and prevalent, such as Clostridium, anthrax, tuberculosis, and Staphylococcus aureus. And these are all diseases that you have some very cool way of evading our immune system, and now we're kind of like restricting its capacity to do so. And with this restriction, we're actually seeing kind of like, I guess, like a new way of thinking about handling these diseases, because we're trying to avoid them from becoming prevalent and large again. So this is just going to be like my final slide that I'm talking to you guys. So take this example that I'm showing you. We're talking about resistance and the fact that this is something that's evolved, and there's another group that was very curious about this happening with these antivirulence compounds. So we have Candida albicans again, and then a compound very similar to 902. And what they tried to do was, in a wild-type version of this pathogen, added a media where it's supposed to form hyphae. And when they measured its capacity to basically form hyphae with a long generations of these pathogens growing in this compound, what they saw was that it was able to kind of like remain like in like this steady like yeast state without like rates of evolution. And I don't have a pointer, and I don't actually have a graph, but as a microbiologist, traditionally how these drug resistance tests look is that you see like nothing for like a very long time, and then all of a sudden it's kind of like this exponential growth of resistant cells. And seeing this plateau actually says like a lot to us. And you can even see that after day 14, when they quadruple the dose, there's still no levels of resistance that are being reported. And what is really also exciting is that this strain It's kind of like a drug-resistant version. It's actually one of the most lethal versions of this pathogen that we have, and it tends to mutate very quickly. We're seeing the same thing here. Scientists actually realized that the case of these heavy selection pressures that drugs have on these microbes are what is driving this evolution, which, I mean, makes sense and is like very like intuitive, but it's not something that we really cared about that much before because we didn't really have an alternative. And now here we're seeing with compounds that inhibit Candida albicans' capacity to actually become virulent and evade the immune system, we can get these cells in and just like treat them. And what is overall, again, like really cool, just to like paint a story for you guys, we learned that communicable diseases are things of the past, and these diseases tend to be ones that have some way of evading our immune system or fighting it in some way. And then we use drugs, and now drugs aren't working, but now we have a way to kind of like resensitize them to our immune system. And this is what's really great, this is like what's new, and we're really hoping that this can curb those death tolls that we're estimating to come in 2050. And I just want to take a moment and thank Tammy for having me here, and you guys just listen.